Many of the ideas we'll explore were once dismissed as speculation, before becoming the foundation for future understanding. This series is for those who stay curious anyway. Welcome to the Holoverse.
Cut a flatworm in half, and the piece left without a head does not simply wait to pass away. Within days, it grows one back and becomes a whole worm again, built entirely out of the fragment of itself. Biology has understood this for a long time. It is one of the clearest examples we have of a living thing reading its own instructions and rebuilding what was lost. But those instructions are not always read the same way twice. Every so often, the same genome inside the same kind of worm tells a different story than the one you would expect, and researchers are still working out why. This is where that story gets stranger, and where we pick it back up. Last time, we asked how DNA remembers, how a molecule holds a record across generations, long enough to outlast empires, long enough to still carry a war, and a crossing that history itself forgot to write down. We closed on a question. Now that we can read the library, and now that we're learning to write in it, what will we choose to add? But there's a harder question underneath that one. Every cell in your body carries an identical copy of that library, word for word. And yet, one of those cells becomes a neuron, and another becomes a piece of your liver, and another becomes a piece of your immune system that will spend your whole life recognizing one specific shape of virus or bacterium. Same book, different stories. So here's the question. If every cell holds the same instructions, what decides how each one gets read? And who is doing the deciding? DNA doesn't just remember. It also has to be read, interpreted, switched on and off, and talked about between the parts of a body that all supposedly agree on what the book says. This is part 2. This is what the library never wrote down. In 1961, two scientists working in Paris, François Jacob and Jacques Monod, published a paper describing something in ordinary gut bacteria that changed how biology understood the gene forever. They found a cluster of genes that only switches on when a specific sugar and lactose is present in the cell's surroundings. A repressor molecule sits on the gene like a hand over a switch, and lactose is the only thing that moves that hand. The gene itself isn't just sitting there waiting to be read. It's being read on command by machinery that answers to what's happening around it right now. The discovery built on earlier work with author party won Jacob, Monod, and their colleague André Louf the Nobel Prize in 1965. This is established foundational molecular biology. If part 1's DNA was a library, then this is the librarian. The same shelf read differently depending on who walks in the door. And here is something you might find fascinating. In 1942, decades before anyone had ever seen the physical structure of a gene, a biologist named Conrad Weddington drew a marble rolling down a landscape of hills and valleys, pulled to one valley or the other by strings attached to pegs. He was trying to picture how a developing cell chooses what to become. He didn't know what a gene was made of. Watson and Crick's paper on DNA structure was still 11 years away, and the picture still basically works today. He even gave the whole field its name, epigenetics. Not everyone agrees this is even the right way to talk about it, and it's worth paying attention to. The philosopher and biologist Susan Oyama has argued for decades that calling genes a stored program that gets read smuggles in a false picture. In her view, development isn't a book being executed. It's an ongoing, genuinely interactive construction between the genes, the cell, and the environment, with no single layer in charge. That's a real, live disagreement inside the field, not a settled matter. And it's one worth taking into consideration before we get too comfortable with switch as the only word for what's happening here. If a cell can flip a switch on a gene without changing the letters underneath it, an obvious question follows. Can that switch setting itself be passed to the next generation? Not the sequence, the annotation in the margin. In some plants, roundworms, and fruit flies, the answer is established. Yes, it happens. Whether it happens in humans is genuinely actively disputed, and this is where we have to slow down and be careful. In a previous episode, we covered the strange edges of inherited memory, about how mice were trained to fear a scent, and passing that fear to pups who had never smelled it themselves, and how monarch butterflies were finding their way to a forest that none of them had ever seen. Here's where that thread continues now. In 2013, Brian Diaz and Carrie Ressler at Emory University conditioned male mice to fear acetophenone, a chemical described by the original researchers and by science writers since as smelling something like cherry blossom. They paired that smell with a mild shock and then bred the mice. Their offspring, who had never smelled that odor and never felt that shock, became startled specifically at that scent and showed a physically enlarged version of the brain structure that was tuned to detect it. The researchers also reported a chemical change at the corresponding gene in the father's sperm, and that's the published, peer-reviewed finding. Whether it proves inherited memory in any mechanistic sense is yet to be settled. A Columbia molecular biologist, Timothy Bester, has argued the specific gene in question doesn't even have the chemical structure the proposed mechanism would require. Other researchers call it among the most rigorous demonstrations of its kind in a mammal. And both of these things are being said right now by working scientists about the same data. The monarch butterfly carries its own version of this question, and it's worth keeping the record straight here too, because the popular idea that monarchs inherit a precise map to the very grooves their ancestors wintered in reaches a little further than the evidence does. No single monarch ever completes the full round trip between the eastern United States and the overwintering forests of central Mexico. It takes 3 to 5 generations to finish that journey every year, and the butterflies that make the long return leg south have never made that particular journey in their own lifetime. They still find their way, though, by using a sun compass that's built in their bodies before they were ever hatched. This is established and genuinely remarkable, and definitely not in question. What's less certain is whether they also inherit that precise map or only a general sense of direction. A 2022 study shifted the magnetic field around migrating monarchs and found that they kept heading for the same sites anyway, suggesting the inherited compass may be about direction and not necessarily a pinpoint of a memory of place. That part stays an open question. And there's one more thread here, close to home. The Dutch famine of 1944 to 1945, known as the Hunger Winter, struck the German-occupied Western Netherlands at the end of the Second World War, when a transport blockade combined with one of the harshest winters on record. People born to mothers who were pregnant during that famine, decades later, showed measurably different rates of a particular chemical mark on a gene involved in growth, along with a higher rate of certain adult illnesses, at least compared to their own siblings who weren't exposed. That finding in that first generation is well established and remains one of the strongest human data points we have for this question. Whether that mark reaches all the way to the grandchildren is thinner, newer, and still being followed as that generation ages. The honest answer right now is that we don't fully know yet. So, can an experience, not just a sequence of letters, leave a mark durable enough to cross into the next generation? Some of the evidence says maybe. None of it says yes for certain. That's where the science stands. Some conversations don't happen between generations. They happen between individuals, in real time, and some of the loudest ones happen between organisms with no brain at all. In the 1970s, researchers Woodland Hastings and Kenneth Nielsen described a marine bacterium, Vibrio fischeri, that lives packed inside a small squid's light organ. Each of the bacterium releases a tiny signaling molecule into the fluid around it. Alone, that signal is too dilute to matter. But once enough of them have gathered, and enough of that molecule has built up, something clicks, and the entire colony switches on its bioluminescence together. They are, in effect, counting themselves before they act. Bonnie Bassler later found something stranger still. Many different species of bacteria share a second signaling molecule, on top of their own private one, a kind of chemical language that crosses species lines entirely. That's also established. Whether communication is the right word for any of this is a real and ongoing argument. In 2002, the microbiologist Rosalind Redfield published a challenge to this framing, arguing that what looks like a deliberate group coordination could just as easily be each individual cell privately measuring how fast its own signal is drifting away, with no cooperation implied at all. The chemistry isn't in dispute. Whether it deserves the word conversation, on the other hand, is still being discussed today. And here's a pattern worth naming directly, because once you see it, you'll notice it again before this expedition is over. Between the 1930s and the 1940s, a Yale anatomist named Harold Saxton Burr built his own vacuum tube voltmeter and spent decades measuring faint electrical fields around living things like plants, salamanders, and people. He proposed that these fields, which he called L fields, might be the organizing blueprint of the living body, guiding development and healing before any visible change appeared. Burr personally did the measurements and published them. But the bigger claim, that voltage itself was the blueprint of life, was considered too close to vitalist thinking for most of the mainstream biology to take seriously then and for decades afterwards. Then, starting in the 1990s, a Tufts developmental biologist named Michael Levin circled back to this idea with much better instruments. His lab found that by briefly interrupting the electrical conversation between cells in a cup flatworm, that same regeneration we opened up with causes it to regenerate a second head instead of a tail. And if you cut that two-headed worm again, you would get more two-headed worms. Indefinitely, with the genome never touched. The tissue is building toward a target shape, and that shape is being carried by a voltage pattern, not by any change in the DNA. Decades later, in 2020, Levin published a paper explicitly revisiting and crediting Burr's once dismissed work. Levin's further claims that cells and tissues might have their own basic decision-making or collective intelligence remains genuinely debated among biologists. Now here's the pattern doing it again, in a completely different corner of biology. In the 1920s, a Russian embryologist named Alexander Gruvich reported that one cluster of onion root tip cells seemed to nudge a separated neighbor into dividing faster. This was through a radiation he called mitogenetic rays. The claim was controversial in his own time, and it stayed that way for 50 years. Then, in the 1970s, a German biophysicist named Fritz Albert Popp built equipment sensitive enough to detect single photons. He confirmed that every living cell he tested, like seeds, sprouts, and animal tissue, genuinely gave off an extremely faint light in total darkness. That base finding, called biophoton emission, is real, and it's been independently replicated since. What's still unproven and treated with real skepticism is Popp's bigger claim, that the light is organized with laser-like precision and carries meaningful information from cell to cell. Researchers who study it today are adamant that this part remains, in their own words, still under investigation. An idea framed as too subtle, too vitalistic for its era, and dismissed for decades, then given a much narrower, more careful confirmation once the instruments finally caught up, without necessarily proving the biggest version of the original claim. Burr and Levin, Groovich and Pop. Two completely separate stories and the same shape twice. Worth noting and worth watching for when it happens again. I would like to take a moment to share that if you enjoy wonder and curiosity, we built an Observatory Foundation. It goes beyond your normal premium Vault template by making you feel at home while you work and is gentle enough to leave you with energy at the end of the day. It's customizable so it could be tuned to how you live and not a fixed shape that everyone has to fit into. I'll leave the link in the description. Now let's keep going. Some of this conversation never stays inside a single organism at all. Your immune system is a memory built entirely out of communication. In 1957, the immunologist Frank McFarland Burnett proposed that each of your immune cells carries its own uniquely shaped receptor, made by cutting and rearranging its own DNA before it ever met a pathogen, counting on a shape it might need someday. When a matching threat finally shows up, that one cell's lineage multiplies by the thousands, and some of those copies stick around afterwards as memory. The whole system runs on a constant chemical back and forth between cells, called cytokine signaling, coordinating how big or small a response should be. All of that is established. Signals like this aren't limited to mammals either. In 1873, at Charles Darwin's own urging, the physiologist John Burdon Sanderson recorded something nerve-like, an electrical action potential inside a Venus flytrap. Darwin, who mailed him the specimens, called it the most wonderful plant in the world. It was the first proof that this kind of signal isn't unique to creatures with nerves at all. And underground, in forest soil, fungal threads physically connect the root systems of separate trees, and resources measurably move through those connections. That much is established. What's genuinely contested is how far those connections go. A major 2023 review found that the popular narrative that trees are talking to each other overstates both how these networks are structured and what they do forest-wide. And that mapping exists for only a tiny fraction of the world's tree species. The scientist whose original work the popular story of the Wood Wide Web is built on disputes that framing and stands by the broader picture. Both sides agree the physical connections are real. They disagree about what those connections mean. Before any of this information could be measured, there were historical records that describe similar ideas. Waddington's landscape and Darwin's flytrap are both documented cases of someone correctly intuiting a switch or a signal decades before the instruments existed to prove it. But there's an older and very different kind of record too, one that sits outside empirical science entirely, and we want to be careful not to blur the two together. Traditional Chinese medicine describes qi flowing through channels called meridians, connecting the body's organs. Mainstream biomedicine has looked for meridians as physical structures and found no evidence supporting them. What's interesting is even while rejecting meridians as real anatomy, mainstream clinical medicine has adopted acupuncture as a recommended treatment for certain conditions like nausea and pain management. Our Vedic and yogic tradition describes something similar in its own language. Prana, a life force flowing through some 72,000 nadis, which are channels layered over but distinct from the physical nervous system. Hermetic philosophy holds that the body mirrors the cosmos itself, as above, so below, an idea the Renaissance scholar Marsilio Ficino revived centuries later. And in the 20th century, thinkers like Henri Bergson and William Reich each proposed their own organizing life force, coordinating a living body from within, distinct from ordinary chemistry. Mainstream science regards those specific mechanisms as superseded, and in Reich's case, as pseudoscience. None of these traditions predicted quantum sensing, or bioelectric fields in any technical sense. No direct lineage connected them, but independently, across wildly different centuries and toolkits, people kept arriving at the same basic intuition, that a living body is a network, carrying something unknown between its parts, long before anyone could point at a molecule and prove it. That resonance is only thematic, but it's still worth noticing. Part 1 asks how DNA remembers. Part 2 asks something almost the opposite. Given that memory exists, who's reading it, moment to moment? And what happens between the reading? What we found wasn't one answer. It was a body full of arguments still being had in real time. A repressor protein that decides, mid-sentence, which part of the same book gets spoken aloud. A scent that maybe, maybe not, leaves a mark on a grandchild who was never there to smell it. Bacteria that either coordinate on purpose or just happen to look like they do. A worm that grows the body its voltage tells it to, not the one its genome would predict. A faint glow in the dark that every living cell gives off for reasons that are only half understood even now. And twice, we found the same pattern repeating. An idea considered too delicate, too old-fashioned, too vitalistic to take seriously. Set aside for decades. Then, someone with a sharper instrument circled back to it, confirmed it, not entirely but partially. Burr's voltage, Gruvich's light, both dismissed in their own era, both partially vindicated in someone else's. It makes you wonder what else has already been noticed once by someone with the wrong tools at the wrong time and quietly shelved, waiting for an instrument that hasn't been built yet. We keep drawing a hard line between the archive and the argument. DNA is supposed to be the fixed part, the part that just sits there and gets copied. And communication is supposed to be the moving part, the improvisation layered on top. But that line got blurrier the further we dove. The switch settings themselves might be part of the record. The voltage pattern might be part of the instructions. The conversation between cells might not be separate from the memory at all. It might just be what memory looks like while it's happening. Every tradition we found, thousands of years apart, independently arrived at some version of the same picture. A living body as a network, carrying something unknown between its parts. None of them could prove it because none of them had the instruments. But they kept noticing it anyway, in body after body, culture after culture, long before anyone could point at a chemical and say, there, that's the signal. Maybe that's the thread connecting everything. Not a hidden mechanism, but just a very old habit of noticing that living things seem to be talking to themselves, long before we had any way of listening in. The library remembers. That was part 1. Now, it turns out the library also listens, answers, and sometimes argues with itself, cell to cell, in a language we've only just started to learn how to overhear. What else do you think it's saying right now that we don't yet have the instruments to hear?
If this kind of exploration resonates with you, you're welcome to join me here by pressing like or subscribe.